Welcome to The Explainer. Today, we're diving into a really powerful new suite of AI tools called ImmuneBuilder. It is totally changing how we see, understand, and even engineer the tiny defenders in our immune system. So let's get into how AI is decoding our body's own defense force.
Thanks to next-generation sequencing, we are just swimming in data. We have the genetic blueprints for literally billions of different antibodies and other immune proteins. It's this massive, and I mean massive, library of biological code, and it's growing at a totally incredible rate.
But here's the catch. Knowing the sequence is one thing, but it's kind of like having the blueprint for a skyscraper without knowing what it actually looks like. To really get how these proteins work, how they grab onto a virus or cancer cell, we need to see their 3D structure. And that has always been the major bottleneck, until now.
So let's start by getting a really clear picture of the core problem these new AI tools were built to solve. It really all comes down to a fundamental mismatch in modern biology.
See, on one hand, you've got this fire hose of sequence data. It's vast, and we get it incredibly fast. But on the other hand, figuring out the actual 3D shape of even one of those proteins using experiments, it's painstaking, it's slow, and it is very, very expensive. We've desperately needed a bridge between these two worlds.
Now, what's so cool about these proteins are their business ends, these things called complementarity determining regions, or CDRs. You can think of them as these super flexible loops, especially one called CDRH3, that act like little grappling hooks, latching onto specific threats. Being able to predict the shape of these loops accurately, that's basically the holy grail for designing new medicines.
Okay, so with that problem laid out, the need for fast, accurate structural data, let's check out how researchers are tackling it with this awesome new toolkit, the ImmuneBuilder suite.
ImmuneBuilder isn't just one single AI model. No, it's a whole specialized toolkit. You've got ABodyBuilder2 for antibodies, NanoBodies for these unique things called NanoBodies, which are smaller and more stable, and TCR-Builder2, which is for T-cell receptors. Those are absolutely crucial for our immune cells to recognize and attack infected cells. Each tool is an expert, trained for its one specific job, and that specialization is a huge part of its power.
So how does this AI actually do it? How does it take a simple string of letters, a protein sequence, and turn it into a complex, working 3D structure? Well, the process is actually remarkably elegant.
It follows this four-step pipeline. First, you just feed it the sequence. The AI then spits out a little group of 4 slightly different structural predictions. Then, and this part is really clever, it picks the one structure that's closest to the average of all 4. It's a great way to make sure the prediction is robust. And finally, that best guess gets put through a refinement step using a physics tool called OpenMM to just polish it up and make sure it's physically realistic.
Now, what's really interesting here is that it's all built on the same revolutionary architecture as Google's AlphaFold-2, but because it's specialized just for immune proteins, it gets to take this massive shortcut. It doesn't need to do something called multiple sequence alignments, which is a really time-consuming step where you look for evolutionary data. That specialization is the secret sauce behind its incredible speed.
Okay, let's get to the proof. Because in science, talk is cheap, right? It's all about the data. So how does ImmuneBuilder actually stack up against the competition?
For this comparison, we're going to put ABodyBuilder2 in a head-to-head matchup, and its opponent is AlphaFold-Multimer, which is pretty much the reigning champion, the gold standard for predicting these kinds of protein structures.
Okay, so this table shows the accuracy for that all-important CDRH3 loop. It's measured in something called RMSD. Basically, it's the average distance between the predicted atoms and the real ones. A lower number is better. And check this out. ABodyBuilder2 hits 2.81 angstroms. That's state-of-the-art accuracy, and it's even a little bit better than AlphaFold-Multimer.
And this is where the story gets really transformative. Let's talk speed. AI Builder 2 can generate a full, high-quality antibody structure in about 5 seconds on a single GPU. 5 seconds! That's practically instantaneous.
Now, compare that to the gold standard. Even a modified, faster version of AlphaFold-Multimer takes around 30 minutes to do the exact same job.
So, the crucial point here is this. We're not talking about a small improvement. This is a speed-up of over 100 times. This is what takes structural prediction from a specialized, one-off task to something you can do on a massive scale. You can finally analyze huge datasets of immune proteins. It's a total game changer.
But hey, speed is useless if the results are junk, right? Well, crucially, unlike some of the other fast models out there, ABodyBuilder2 produces physically realistic structures.
The atoms are all oriented correctly in 3D space. The quality is right up there with what you'd get from actual experiments.
So with that kind of performance, top-tier accuracy at a tiny fraction of the time, let's explore where a tool like this can really make a difference, and just as important, what its current boundaries are. The applications here are just so exciting. This can speed up the design of new antibody drugs, help us engineer better T-cell receptors for cancer treatments, and let researchers screen enormous immune repertoires to find the best candidates for new vaccines and therapies. The potential is just huge.
But it's really important to know its limits. This is a specialist tool, not a jack-of-all-trades. It's only for these specific immune proteins, not for general ones. It has a max sequence length, and, its predictions for some really long and wacky CDRH3 loops might be a little less reliable. So, when you put it all together, what does a tool like ImmuneBuilder really mean for the future of biology and medicine?
Well, to prove its power and their commitment to open science, the research team didn't just build the tool. They actually used it to predict the structures for over 148,000 unique antibody sequences, and then they released all of that data to the public, completely for free.
And this quote from their paper just nails why this is so practical. It's not just about speed and accuracy. It's the fact that the tool gives you an error estimate for each prediction. That lets researchers confidently filter their results, making it a truly useful and reliable tool for actual lab work.
This massive leap in speed and specialization really opens up a whole new world of possibilities. It really makes you wonder, with AI tools like this predicting biology at this kind of speed, what's the next great scientific frontier we're about to unlock?